In the last module, we talked about the importance of ATP and how it is essential not only for exercise, but also for life. However, our bodies do not store large amounts of ATP. We don't just keep this huge reserve that would allow us to just burn our internal reserve stores for hours and hours and hours. Indeed, we have about enough ATP in the cell to maintain exercise for a couple of minutes. So if we didn't have a way to replenish that ATP, then during exercise, during high-intense exercise, we would literally fall over and die. Now, while some of you may be thinking, I've experienced that, and you've worked out really hard and just felt like you could die, that wasn't because of a depletion of ATP. So this doesn't happen. And why doesn't it happen? It doesn't happen because we have ways of making more ATP in the cell. We have three metabolic pathways that, again, are going to take our foodstuffs and convert them, our carbohydrates, our fats, and a little bit of protein, but pretty minimally, and convert that into ATP so that we can continue exercising. Right? So the first one that we'll talk about in this section is the phosphocreatine system. Phosphocreatine system is the simplest of all systems. It takes this molecule phosphocreatine and essentially makes an ATP out of it. We'll go in more detail in the next slide. The second one that we will also cover in today's lecture is glycolysis. Glycolysis is the breakdown of the simple sugar, glucose, the monosaccharide, the only one that we can convert, if you'll remember from lecture, and it converts it into ATP and also two products that can further on be metabolized into pyruvate or lactate. The last one is oxidative phosphorylation, and that will be the subject of our last module in this unit. So if we focus here on the phosphocreatine system and glycolysis, we call these two systems anaerobic. When we say anaerobic, what we really mean is they do not require oxygen in order to make ATP. Right? Because of this, they occur in the sarcoplasm of the cell. This is where all the enzymes are located, and they are going to be used to produce ATP relatively quickly. On the other hand, oxidative phosphorylation, as the name implies, is that it is oxygen dependent. We require oxygen in order for this to happen, and therefore we call it aerobic metabolism. Because of this nature, it occurs in the powerhouse of the cell in mitochondria. Right? And you may be thinking to yourself, how come, since we're making ATP in the phosphocreatine system and glycolysis, why isn't the sarcoplasm also called the powerhouse of the cell when we call the mitochondria that? Well, as we'll learn, is the sheer volume of ATP that we can generate is much higher in the mitochondria than using either the phosphocreatine system or glycolysis. Right, so we'll highlight that's a little bit of a negative. However, the rate of ATP production is really fast. So let's jump into those two systems and discuss the pros and cons of each. So the first is the ATP-PC system. So I'll just say this, there are lots and lots and lots of names for this system. Everyone has kind of their own vernacular that they like to use. So some people, I'm partial to the phosphocreatine or PCR system. We also have the PC system. Here we have the ATP PC system. We also have the phosphagen system, which I won't write out. We will also speak out here, it's spoken of as the CK reaction or creatine kinase reaction based on the enzyme that does it. And I think that that locks them up. So again, lots and lots of names to just make everything nice and confusing for you. Okay, so what is the phosphocreatine system? The phosphocreatine system essentially is the simplest of all metabolism, right? It's called substrate level phosphorylation because we take our substrate, ADP, and we add this phosphate, relatively simple. We add the phosphate here, right? It gets added there. So essentially ADP is phosphorylated onto and becomes ATP, and then we have our creatine molecule left over. Again, very simple substrate level phosphorylation. We have our substrate, we phosphorylate it, we change a D to a T, ADP becomes ATP. So this energy bond is converted from ADP to ATP, and the enzyme that does that is the creatine kinase reaction, or CK. Right? And the reason that this is such an important system is that it is extremely rapid method of ATP production. It's simply rapid because it's one single step. All we're doing is taking ADP and adding a phosphate group to it using the enzyme creatine kinase to make ATP. Very simple, very fast, and adds a lot of ATP to the system very quickly. So therefore, we're going to rely heavily on this system at the onset of exercise and during short term, less than 5 seconds usually, are going to be really predominantly based on the ATP phosphocreatine system here, right? So things like sprinting 50 meters or high jumping or rapid weightlifting is going to really involve heavy input from this phosphocreatine system to replenish the ATP that we are using in our muscles. However, one of the big negatives of the phosphocreatine system is that we just have a small amount of phosphocreatine in our muscle cells. Right? So we have limited space and limited amounts of this protein in the stores, and so ultimately we have a limited capacity to produce ATP via this system. Right? In the perfect world, we would have an unlimited store and we would never have to worry about using oxygen. We could just use all of that. But that's not the case. Very small amount of phosphocreatine system, so we can only maintain, again, this system for orders of magnitudes in the seconds, not in the minutes. And then after that, we start to deplete the system. So if we take one more look at phosphocreatine system, so the reason that we need to do this is if you kind of compare, I have these up here, so if you'll compare the shape or the structure of these molecules, so you'll see. So we'll just highlight this one here. So this inorganic phosphate here is a phosphate group bound to 4 oxygen molecules. In the phosphocreatine system, you'll see the structure is slightly different. So this is the reason that we can't just use, myosin ATPase or some other enzyme couldn't just cleave this phosphate here, and we would skip the entire step of having to convert ADP to ATP, right? We, in theory, could just take the phosphate group off of phosphocreatine directly, whatever enzyme needed it. So, for example, myosin ATPase could come in, cleave this, and use that high energy stored in order to perform a power stroke in the cross-bridge cycling platform. But since these structures are not exactly the same, we don't have the ability to do that. So we can't directly cleave the inorganic phosphate for that. So then we have that issue of specificity, as we learned in the last lecture, of enzymes are only particular to one single structure and function, right? And in this case, the enzyme that is perfectly suited to handle this phosphocreatine molecule is, again, creatine kinase, or CK. So again, one more look at the phosphocreatine reaction. Again, very simple. So we have our creatine bound to our inorganic phosphate, otherwise known as phosphocreatine. Creatine kinase is going to use the energy to produce an ATP molecule by adding the inorganic phosphate, and then some enzyme in the body, you name it, can then use this ATP, right, to break down, store this high energy bond, also create a hydrogen, and have that energy to perform some type of reaction in the cell. I'll also note that to you, though, that creatine kinase can actually go both ways. So the same enzyme is responsible for taking creatine and adding energy in. So we add energy in here, and adding this organic phosphate back to the creatine molecule to essentially make it ready to go again and create the phosphocreatine molecule. So creatine kinase does both. It breaks down creatine and phosphorylates ATP, as well as phosphorylates creatine and gets it into the phosphocreatine state. So how does this system work and perform during exercise training? So is there any way that we can enhance the system? So this is interesting. I like that the book tackles this. However, sometimes it's always not super obvious, right? So there isn't a ton of work in this area, and the problem is that the results are a little bit of both, right? So we see in some studies we see increases in the major enzyme of this, which is of course creatine kinase. If we are able to increase creatine kinase, then of course we would generate ATP faster and be able to generate ATP in a much more powerful response, so that we would be, in theory, faster or stronger in those very short-term duration exercises that the phosphocreatine system is important for. Also, there's a few studies that say with anaerobic training that some people can actually see an increase in the amount of ATP, total ATP available in the cell, right? So we can increase ATP and give us a little bit of a boost in, kind of the capacity in which to create energy. And then we can also increase just by training the amount of phosphocreatine. Of course, it makes sense here. I have it that aerobic training isn't going to do this, right? The only type of training that is going to do this is this high intensity, really short bouts, right? Anaerobic training to really stress that phosphocreatine system. So it's still unclear. I can't say that I can't go out and encourage you to say this is definitely possible, but there is some literature out there to suggest it, and I'm glad the book brings it up. I think that the point is that if either of these adaptations occur, then they're ultimately going to improve performance in any of these really short duration events. Second is, do we deplete phosphocreatine? So depletion of ATP and depletion of the phosphocreatine system do not seem to be a primary cause of decreased muscle force or fatigue. Indeed, ATP concentrations stay pretty constant. And as you can imagine, the muscle cells are really honed in on keeping ATP relatively constant during high-intensity exercise. Yeah, we'll see the concentration drop some, but it'll stay at the same concentration for a really long time unless it's a really high-intense exercise. And again, that's because if we run out of ATP, then the cell will die. However, phosphocreatine system, of course, we can deplete that. We can get all the phosphates out and be left only with creatine in this situation, but that doesn't really seem to be the case. And so what we really believe is that fatigue is caused by acidity, again, from the hydrolysis of ATP and production of the hydrogen ion during that process, as well as a little bit of accumulation from inorganic phosphate from the hydrolysis of ATP. Both will alter the acidity of the cell. But it is possible to completely deplete your phosphocreatine system, and in this case, then some type of recovery period is needed in order to provide energy, right? We need to put energy back into that system to rephosphorylate phosphocreatine system, and we need the aerobic system to do that in order to generate that amount of ATP. So any type of interval training or some really intense anaerobic sports like powerlifting, that this may come into play, you need to kind of think about some type of rest and recovery period shortly after the bout to prevent that, and also providing some carbohydrates in the diet in order to provide substrate for that energy. Right. That closes out the phosphocreatine system, and the last section we'll do is glycolysis.